This chapter covers a lot of material, cancer, DNA synthesis, mitosis, and meiosis. So let's start with some important concepts about DNA and DNA synthesis, and then we'll move to the cell cycle before covering cancer. Remember that the nucleus of the cells holds DNA, which is the genetic material of life. In its normal state, this almost looks like a big bowl of spaghetti. This loose form of DNA is termed chromatin. Chromatin is made up of the DNA double helices, and they're wrapped around proteins that are called histones. When a cell is getting ready to duplicate its DNA, the DNA condenses down into a chromosome. Each species has a particular number of chromosomes. This is the full or diploid number of chromosomes. The chromosomes are arranged in pairs. So this is a drawing of a karyotype or a magnified photograph of chromosomes. So this is a human male karyotype. There are 22 pairs of autosomal or non-sex chromosomes, so that totals 44, plus an X chromosome and a Y chromosome, which gives us a final number of 46 chromosomes. All humans have 46 chromosomes. The pairs of chromosomes that match up are called homologous pairs. In this picture, there are 23 homologous pairs. We're going to talk about two types of cell division in this chapter, mitosis and meiosis. Mitosis is the one that is the normal cycle for your body as you grow and as you heal from injury and disease, as you're making more cells. In this video, I'm primarily going to discuss mitosis. In mitosis, DNA is replicated and a parent cell divides into two identical daughter cells. So for a human, the parent cell would have 46 chromosomes, and the daughter cells would have 46 chromosomes. In meiosis, DNA is replicated as well, but the parent cell divides twice, resulting in 4 daughter cells that each contain half the number of chromosomes of the parent cell. For a human, the parent cell would have 46 chromosomes as usual, but the daughter cells after meiosis would have 23 chromosomes. These cells are termed haploid because they have half the number of chromosomes of the normal diploid organism. These are gametes, so sperm and egg cells, and they're used for reproduction. Meiosis only takes place in the gonads, or the sex organs. When DNA is replicating prior to cell division, the double helix splits in half up the middle. New nucleotides are added to each side of the original parent molecule according to the base pairing rules that we've discussed previously. The enzyme DNA polymerase, which isn't really shown in this picture, moves along the length of the unwound helix and helps bind incoming nucleotides to each other on the newly forming daughter strand. This results in 2 daughter DNA molecules. Each one of them is composed of a strand of old DNA and 1 strand of new DNA. Let's talk about mitosis and the cell cycle first. The cell cycle is an orderly series of events that takes place between the time a eukaryotic cell divides and the resulting daughter cells divide. So it is one generation of a cell. This does not involve meiosis, which is a specialized event that only takes place in the gonads. The cell cycle is divided into 3 general phases: interphase, mitosis, and cytokinesis. It is very tightly controlled. There are checkpoints during the process that can put a halt to it if something goes wrong. Interphase has 3 phases. In the G1 phase, the cell grows larger as the organelles duplicate in preparation for cell division. In the S stage, the DNA in the chromosomes replicates. In the G2 stage, proteins are synthesized that will help drive mitosis to completion. When chromosomes have replicated, they are called sister chromatids, and they are joined together at the centromere. In this picture, the yellow and blue chromatids are identical as far as their genetic material is concerned. Following interphase, the cell enters into mitosis. Mitosis is divided into 4 phases: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes have already duplicated and are composed of 2 sister chromatids that are held together at a centromere. Microtubules form and grow and ultimately form the spindle, where they radiate out from opposite ends or poles of the cell. The nucleolus disappears and the nuclear envelope fragments. This is a microscopic view of plant cells undergoing prophase. You can see both early and late prophase here. The chromosomes in the nucleus have condensed down and the nuclear envelope is disappearing. So the chromosomes are starting to separate out. Look how different that appears compared to the cells around these cells that are in prophase, where the content is very indistinct and you cannot see the chromosomes at all. In metaphase, the replicated chromosomes align across the middle, or equator, of the cell. The microtubules line the chromosomes up in a single file line across the middle. Here you can see those chromosomes lined up in the middle of a cell during metaphase. In anaphase, the centromere splits and the microtubules shorten to pull each sister chromatid of a chromosome to opposite poles of the cell. This is anaphase in an animal cell. You can see the chromosomes are pulling apart from each other. This is the shortest phase of mitosis. It happens very, very quickly. Here's another picture, kind of at the tail end of anaphase. You can see the chromosomes have separated and are moving towards opposite ends of the cell. In telophase, the nuclear envelope reforms around the newly produced daughter nuclei, and the chromosomes revert to their uncondensed form. This picture of animal cells shows telophase as well as the first part of the next process, which is cytokinesis. In cytokinesis in animal cells, a band of proteins encircles the cell at the equator and pinches it off, dividing the cytoplasm in half. Here's a sketch of cytokinesis in animal cells. In plant cells, a new structure or a new cell wall must form between the two cells. A structure called a cell plate forms where the new cell wall is being built. Alright, so let's talk about the cell cycle and cancer. There are some medical terms that you should probably be familiar with. You may come across these at some point in your life. Oncology is the study of cancer. Histology is the microscopic study of tissues. Whenever they're being studied microscopically, a chunk of tissue is embedded in paraffin wax and then sliced very, very thinly, so that it's only about 1 cell thick. That thin slice of tissue is placed on a microscope slide, stained with special dyes, and then looked at through the microscope. Pathology is the study of abnormal or diseased tissue. Cytology is a little different from histology because it involves looking specifically at isolated cells. Your doctor or vet might use a needle to aspirate or collect cells from a tumor, then place them on a microscope slide, stain it, and look at it under the microscope. These cells are no longer in their normal architecture, so interpreting them can be a little bit difficult. Here's a histological slide of the small intestines. You can see the normal architecture of the organ, so it's very easy to see what isn't normal. Histology from a biopsy sample is the most accurate way to diagnose cancer. Pathology is the study of abnormal or diseased tissue. So to know what's abnormal, you have to know normal. Here is a picture of a normal kidney, and here is an abnormal kidney. Gross pathology doesn't mean disgusting pathology, although it often is. It just means something you can see with your eyes, as opposed to microscopic pathology. This is an example of polycystic kidney disease, a very common inherited condition in Persian and Abyssinian cats that can lead to kidney failure at a fairly young age. Cytology is the study of cells. This sample here would have been collected from something, either a mass or an organ or a blood sample, and then put on a microscope, stained, and looked at. This is an example of cancer, a large cell lymphoma. Here are a few more terms that are important to understand. Diagnosis is the process of determining the type of disease present. Also, it can be considered the type of disease present. So you can diagnose a condition. The diagnosis is the disease, or you can work through a diagnosis trying to figure out what the disease is. That term can be used for both of those things. The prognosis is the expected outcome from the disease and or the treatment of the disease. So if you have a sick person that has a pneumonia, the prognosis might be guarded. Guarded meaning that they could recover or they could die from it. So the prognosis is the expected outcome. A biopsy, or taking a biopsy, is removing a piece of tissue from a lesion to study microscopically, typically through histopathology. Here is a picture of how a human might take a skin biopsy using a punch biopsy device where they can lower it into your skin and they kind of twist it around, and then when they pull up a piece of tissue with that. A mass is a collection of abnormal tissue that has borders and is easily identified as different from the surrounding healthy tissue. A nodule is a small abnormal growth of cells, or a small mass. A tumor is a large abnormal growth of cells. And then neoplasia is the technical term for cancer. Neoplasia literally means new growth. In this picture, the nodule is the small button-shaped growth on the ear there on the upper left-hand side, and then the tumor is much, much bigger. So that's the dog below with that bulging out from the side. That is a tumor. Benign cancers, or benign neoplasias, are the best kind to have because they're not malignant, so they're not typically dangerous. They are unlikely to spread to other locations in the body. Malignant cancers, or malignant tumors, are likely to spread to distant sites in the body, and they're very aggressive in growth. Metastasis, or metastatic, involves the spread of a cancer to distant locations in the body, or the tendency of it to spread. Angiogenesis is the creation of new blood vessels to support a tumor. So if you see angiogenesis with a tumor, that is a sign of aggression of malignant tumors, because they are more likely to have spread in the bloodstream if they have a good blood supply. The lungs are a common location for tumors to spread to, and so in this picture you can see in the chest cavity the arrows are pointing to several of the lumps that have come up in the lungs that are spread of cancer from other locations into the lungs. Those are tumors that have metastasized to the lungs. Cancer is a cellular growth disorder that occurs when cells divide uncontrollably. Although there are lots of different causes, most cancers are the result of accumulations of mutations that eventually cause a loss of control in the cell cycle. Your textbook discusses some of the genes that are involved in cancer formation. Be sure to read that section to understand the importance of proto-oncogenes, oncogenes, tumor suppressor genes, and specifically the p53 gene. Diagnosis and treatment of cancer is best done systematically. The traditional and still most widely used method of diagnosis is histologic examination of tissue removed by a biopsy. So here is a picture of an osteosarcoma, but it's of a leg. So this is near the knee, basically. Okay, so just below the knee. And if this was on a dog, the dog would present with a growth near the knee that's very painful. It might be limping. It might not be able to bear weight on that leg at all. So if you took a sample of that growth that you saw in the x-ray and looked at it histologically, you would see that it is very abnormal. All of these strands and curly amounts of cells should not be there. They're not normal in bone. A fine needle aspiration biopsy, also called a fine needle aspirate, produces a sample consisting of both small tissue fragments and isolated cells. It can often be done on location, so right there in the office by a trained doctor or veterinarian, but the accuracy of this is really dependent upon the skill of the person collecting the sample and also the person reading it under the microscope. So if we did a sample of that growth on the leg near the knee, you would see something like this, and these are cancerous cells, and they're very consistent with cells you would see with an osteosarcoma. Characteristics we look for that are characteristics of malignancy include a high rate of mitosis, so that's called a high mitotic index. That means the cells are dividing very rapidly. If they have various cell shapes and sizes that are not consistent, that can be a sign of malignancy. Also, if they have abnormal nuclei and nucleoli, that can be a sign of malignancy as well. If you need to research cancer, there are some reliable websites you can go to. This is a couple of them, and these are the ones I'd recommend going to if you have someone that you know that has cancer and you want to find out more about it.